And then today I cannot stay till the end of your lecture either, because and there we go. So as I said, this is recorded so people can see it also tomorrow and still see it before your class.
No.
So let's go to the first page. Yeah.
Okay,
And full screen. Oh yeah,
okay.
that looks good. Fantastic.
That's
Good.
good.
All right. Great pleasure to introduce Anna Marciniak-Czochra, a good friend and colleague from University of Heidelberg in Germany. Uh, Her work is absolutely instrumental in the study of leukemia and stem cells and stem cell feedbacks and these kind of things. And it's a great pleasure that she agreed to give 2 lectures here in this class, today and on Thursday. Today, unfortunately, I'm sorry, I messed up the time zones and we are 2 hours late. But on Thursday, we will be on time, usual time, 10 o'clock Toronto time. Anna, we are looking forward to your presentation.
Thank you, Thomas. Thank you for the introduction. It's my pleasure to give this 2 lectures today and on Thursday. And since I was thinking about this, which aspects of cancer modeling and dynamics we could cover in this lecture, I decided that we can look at the heterogeneity, but heterogeneity from this perspective of cellular hierarchies. So I will show you different aspects of modeling and model analysis, but also model-based data analysis of stem cell-based systems. And so, okay, we aim at understanding cancer, but somehow first one should understand such system in wild type, in healthy situations, so models of regeneration. And I focus on 2 systems on which my group has been working for many years now. It's blood and then neural stem cell system. So today I will be speaking about blood modeling, blood stem cell modeling, and on Thursday more on neural. And then also today I also would like to show you development of different approaches for modeling depending on the questions. And then somehow we come to motivation of treating high dimensional problems. And then on Thursday also I would like to show you some new challenges and some new mathematical analytical ideas for structured population models. So I speak about structured population models, but in fact, what I want is pretty general. I want to describe dynamics of populations which exhibit heterogeneity. And of course, all populations exhibit heterogeneity on very many levels, but we are interested in this heterogeneity which is functionally relevant, and in particular functionally means relevant with respect to the processes we describe. So it is like all populations are has age structure, but if we are interested in processes for which age is not important or not changing parameters, changing processes, that we can neglect it. So we are looking here at this functionally relevant heterogeneity. And of course, we can think about such structures of heterogeneity in very different ways. We can think about discrete structures. We can think about continuous structures. I will show you some example of this. And of course, we have structures, this internal structures of the system on different scales and in different dimensions, and then they are also often additionally regulated and changing in time. And if we think about applications, of course, applications are everywhere. I'm interested in mostly in cell and developmental biology and related also fields of mathematical oncology. And maybe before I go to simple model and all these developments, I would like to show you the striking example of heterogeneity, which nowadays is, let's say, one of the main issue in research of most cell biologists, but not only cell biologists, also from clinical perspective and in developmental biology. Namely, incredible advancement of experimental tools allowed people to get information on cell systems which are highly dimensional. These are so-called omics data. You can hear often about multi-omics data. So this are single cell data. They are data describing position or characterization of individual cells. And now depending on which omics it is, it can be genomics, transcriptomics, proteomics. So you may have different measurements. So we look in the context of this, what I will be talking about, one looks mostly at transcriptomics, so looks at transcripts, and then just cells from the system undergo a single cell sequencing. And then you get information about each cell, which is in 500 dimension or more, informations about transcription of different genes. And then there is a lot of machinery of bioinformatics, which takes the most important dimensions from this. So this is like mostly at the beginning some linear algebra tools to find principal component analysis, to find directions in which there is biggest heterogeneity, and then using on the way many different sophisticated and not always very rigorous tools, one tries to visualize such cell systems with respect to heterogeneity. So here, of course, pictures you see in 2 dimensions, but usually the data which are just here visualized are 5, 7 dimensional. And it shows that the system about which one says, okay, they were like stem cells or they were like one type of cells, one see that in these spaces, they are not in one point, they are spread, so they are very heterogeneous. And one of interesting questions for us, for modelers, for mathematicians, is how can we use this data in our process-based modeling, or can we get any mechanistic information from such data? So I showed you now this just to show you mine at the moment main motivation of understanding which mathematical descriptions are good for describing heterogeneity and how we can extend them to deal with high dimensional data and processes in such spaces. So in the next 50 or 60 minutes, I would like to show you first basic mathematical frameworks to describe heterogeneous population, heterogeneous with respect to hierarchical cell position during development. And just we can ask ourselves a question, which conclusions can be driven from such models? So how comprehensive description we need for particular questions arising from analysis of patient data, of experimental data, and if the different frameworks are equivalent. So as I already announced, I will focus now mostly on the blood system. So it's my now prominent example, granulopoiesis. And if I speak about cellular hierarchies, I mean that the system has such tree-like structure of cell lineages when we have some stem cells on the top of it, and the stem cells divide, and after division could be symmetric division or asymmetric division, and they can reproduce stem cells in these divisions. Like in asymmetric, 1 stem cell, 1 more differentiated cell, in symmetric, or 2 stem cells or 2 differentiated cells. And we can go along such lineage and then have more stages, and on each stage the cells can reproduce their own population or differentiate. Theoretically, maybe not in blood or at this stage of understanding of blood, but in neurogenesis, about which I will speak on Thursday, we may have also sometimes continuous transition. So it means that cell differentiate without a need to divide. And we can also have a cell death in such systems. And it is often that cancer, development of cancer is related to mutations of some cells in such lineage. The earlier in the lineage, the worse, as you will see later also from the models. So we will here focus on one of such lineages of blood, so granulopoiesis, white blood cells production, and then related cancer lineage, so leukemia. So we're interested in acute leukemias. And now when I said about the blood, so what is, let's say, advantage of the blood? Maybe before speaking about complexity, blood is a system which is the longest studies in context of stem cells, and it was in fact this system when the concept of stem cells was first introduced by biologists. So it is a relatively simple system because we can neglect physical space. And the question is, do we care at all about physical space in context of stem cell system? The answer is depends on the system. You have, of course, system when it is essential. These are mostly system from developmental biology, so like developmental system, which are still growing. For example, you may have such growing stem cell structures as in this picture. This is organ of a plant, Arabidopsis meristem, when you have stem cells in the middle and the cells divide, they replicate themselves, the structure is growing, and then depending on the underlying signaling system, like chemical signaling, at some stage the cells start differentiating. These are these blue cells here. So the stem cells are surrounded by differentiated cells. Different fascinating system is are systems like whole organs. So here on this picture you have branchia of the fish, and so it looks like such a cam. And then you have stem cells which sit in the niches, here depicted with this A's, and they are just very few stem cells, maybe 7 or 9 stem cells, which can produce the whole system. And then also they divide, they replicate stem cells, but also they produce more differentiated cells, which then somehow move away just by the process of growth, and also then move into the other direction and produce such a structure. And of course, in these systems, we have to care about this organization in space. If you look at the blood or at the neural stem cell system, about which we'll talk on Thursday, you can neglect the space for 2 reasons. One reason is that and of course, it's not that, if we think about blood, we think about blood in circulation, but blood stem cells are sitting in bone marrow. So there is still a physical niche, physical space, but somehow we can treat these systems for many purposes of our modeling as statistically uniform, and then just don't not to care so much about their spatial position. Although looking at the spatial structure of the niche, also for blood, might be an interesting topic. And if we zoom into the blood niche, so it means this niche in bone marrow, you see how complex the system is, or even the more we zoom in, the more complexity we see. It is not only all this heterogeneity of different cell types in our tree, but we have also, and also spatial structure of the niche, but you may also deal here with such problems as migration of the cell, hemotaxis to some signals, and so on. So in fact, one can say that everybody could find in blood system his favorite type of questions for his favorite type of models of differential equations. And this is indeed a very important decision. So I would say methods, models should be tailored to the questions and processes. Of course, cannot be too simple, but also should not account for details which are irrelevant. And now, depending on our choice, we may consider different cell types. We may look more into the cell cycle or more to the chemical or mechanical regulation of the system. Or to cell migration, and depending on this, choose the proper model. So now, how we model the system. Before I show you our models, I'd like to say that the modeling of blood stem cells started in even end of 80s by Michael Mackey, and then there were many groups also in Canada, in Germany, but also in France and in different places working with such models and just focusing on different aspects, such like stochastic transitions or delay causing oscillations and so on. So this is just some prehistory, let's say. And of
So,
course,
Anna, sorry
yes,
to interrupt you, Anna.
yes.
I just want to tell you that we had Natalia Komarova in this class as well, and she talked about cell lineage models and feedback or homeostasis and so on. So,
Yes,
so the
yes,
students have
of course,
seen.
Natalia also. I mean,
Yeah,
it's like,
no, no,
the slide is far, far, far from any completeness. I just wanted to see how old is blood modeling and so on. Yes, so you see
no, no.
now.
I just say this so that you know what the students have seen before.
Okay,
Okay.
okay, sure.
Yeah.
So, okay, so when we now think about the basic aspect of the lineage, we of course see this classes of different cell types. So we have stem cells, we have progenitor cells, and we have just basic processes. So as I said, the cells divide, produce two daughter cells, and with some probability, the daughter cells stays the same as the mother cell, and then 1 minus this probability, they differentiate. So if you look, okay, now after you told me that you are already aware with the work of Natalia, I could here speed up. So if I want to describe dynamics of such a cell population, then I can like consider cell numbers, and then exactly describe these processes, like with P being proliferation rate and AI being self-renewal fraction for this population. And self-renewal can be interpreted, as I said, as probability that the daughter cell is the same as the mother cell, but also can be interpreted on the level of the population. It means the fraction of daughter cells which stay in the same population, in the same compartment. So it shows that in fact, but in such models, we completely cannot at all distinguish between symmetric and asymmetric division, because having only asymmetric would mean the same as having half symmetric self-renewal, half symmetric differentiation. So just our parameter means what is the fraction or what is the probability that the population stays in the same compartment. And if we would describe such an ODE, then of course it would be exponential growth or exponential decay, unless our A's are exactly 1/2. So it's clear that we are interested in a system which are additionally regulated. And the question is how this regulation can take place. So in case of white blood cells, we know that the system is regulated by cytokines. And these cytokines, so one is GCSF. They are produced by the by the niche, not by our stem cells. So we have for such signal a constant production rate, and we can consider that this signal is taken from the system, but by mature cells. one can also consider here most mature compartments if we consider many compartments. And there is also natural degradation. So this is a very simple model of signaling. If we were focused on specific signaling, of course, one would have to work out this model. But for our purposes, it is first just the general question how the system is regulated. And since such a process is, we can assume that this is faster than cell divisions, then we may take here quasi-steady state approximation. So it means we multiply this time derivative with small epsilon, put this or send this epsilon to 0, and then find S as a steady state or quasi-steady state of this equation. And since I want to work more with signal intensity than amount of signal, then also we rescale the parameters, and then we get such signal intensity being here this Hill function of our UN. So it means the more mature cells I have, the less of the signal intensity I have. And just maybe for your interest, I don't want to go here into details. This quasi-steady state approximation for this model can be done in a rigorous way using a Tikhonov theorem. So it is here, yes, the way how you can, let's say, reduce the model and derive the nonlinear model of regulation from a bigger system. So now the question is, okay, first of all, how we will verify if this control is enough and which parameter is controlled. So we, since we want to understand regulation, we need data which shows how the system behaves far from equilibrium. And in case of blood, of white blood cells, we have such data from patients who had chemotherapy. After chemotherapy, this is here patient data. After chemotherapy, their cell levels went down. So the white blood cells were killed. And then you have regeneration process. They get a bone marrow transplant. This is like a small amount of cells which have some markers for stemness. So they include some stem cells or cells from early stages. And then so like they get 10 to the 5 or 10 to the 6 of such cells. And then within 2-3 weeks, they produce 10 to the 9 of mature white blood cells. And the process here, you have data from quite a few patients, and that you see that the process can be pretty heterogeneous in time. Some of the patients have more monotone growth, some have more like kind of a bit damp oscillations or things like this. So these are the data which we will now use. We will also later look at the far from equilibrium processes related to development of cancer or evolution of cancer. But first, just looking at this data, we want to see how which regulation would be sufficient to describe such reconstitution, hematopoietic reconstitution. And then, since we have two essential parameters in the model, these are proliferation rates and the self-renewal fractions. So these are two candidates which we can regulate with our signal intensity. So once it would mean if I don't have enough mature cells, the stem cells which I have divide faster, makes sense. The other means if we don't have enough mature cells, the stem cells or the early progenitors which we have do more self-renewal, produce more of their own population. So even intuitively, the first strategy sounds more efficient, but model shows that it's opposite. If you would allow proliferation rates to be regulated, the process would be far too slow to cope with the real needs. So like you would need 1 year to recover the blood. While a regulation of self-renewal fractions is very efficient. Here with red line, you see regulation of A. And with black, you see regulation of both parameters. Of course, if I allow regulation of both, it's even more efficient, but there is not a big difference. So conclusion from this was that it's self-renewal which needs to be regulated to explain the process, the data. And we can now compare such models to data from individual patients. Here already we divided them into these three groups with more monotone growth and like here more oscillatory or more chaotic, and here just with overshooting. And in fact, you can easily fit the model to all these behaviors with just very small differences in the choice of A parameters. We can also validate model using large patient group. So it seems that such a simple model can nicely describe reconstitution. And now just for view of the properties of this model. So one can look at the linear stability of the system around this steady state. And first of all, on the structure of steady state, I don't show you here computations, but they are, especially if you look at just at two compartments, they are pretty easy computations. And one can see immediately that you may have trivial steady state. So if your self-renewals would be always smaller than 1/2, that everything decays. So this is
Anna, so I interrupt you again. I'm sorry. Which model are you using now? So I got
now I
lost.
use an ODE model. I see. An ODE model.
This one.
So this is one compartment. You can have n compartments.
Mm-hmm.
And with this model, I have the regulation of self-renewal. So my A parameters look like this. So actually,
Okay, and how many eyes do you use? How many compartments?
so now, depending on the compartments, yes. So in fact, this is exactly what I wanted to say. If you have two compartments, everything is trivial for all computations. But even for general model with n compartments, you can prove this general properties that if all i's are smaller than 1/2, then the only equilibrium is 0. It's like then you have really decay in all equations, and the 0 is then asymptotically, even globally asymptotically stable. If A1, if self-renewal of stem cells is larger than 1/2, then we will have a strictly positive equilibrium. If we would have small, sorry, I see I should have put the equations now all together. So if we would have, for all equations, like if on early compartments, A would be smaller than 1/2, then this is negative. And then you can prove that then all these populations would decay to 0. So somehow system would converge to a steady state, semi-trivial steady state, where it starts to be positive for the first population with self-renewal larger than 1/2. The most important for us is this observation that if self-renewal of stem cells is higher than 1/2, then we have this positive, strictly positive equilibrium. And even this is like, even in a simple system, it is pretty tricky, because you can show, of course, local stability can be proven even for n compartments with linearized stability analysis. But if you want global stability, one can prove it. We could prove it only for system with two compartments. And I just show you here this Lyapunov function for this, that you see how tricky it is. It looks pretty awful. And I said that it's now more than 10 years ago when I said I give a prize for global stability for systems with more than two compartments. So because, I spent ages trying to put something to show it. Of course, you see, part of the difficulty is that if you have at least 3 compartments, for 3 compartments, we can prove even Hopf bifurcation. So it means my parameter regime from global stability is also, let's say, must be carefully chosen, because I have to be outside this Hopf bifurcation. But what is interesting, that parameters which are estimated using this real data from patients are far away from the Hopf bifurcation point. So somehow one can discuss when this Hopf bifurcation here would be applications relevant and so on. but essentially it is not our problem at the first glance. Okay, so this is the structure of our system. And this is the system now a little bit to more general perspective. This is the system about which I call it like discrete structure, so that we have this finite set of compartments and discrete transitions between the set. And in fact, they are discrete because you may transfer to next compartment only during a division process. So they are punctuated by division. One can think now about modeling this process in a continuous way. And in fact, when we did this very first models more than 10 years ago, you didn't have all the single cell data showing heterogeneity. So biologists were completely convinced that everything is compartmental and discrete. But there were quite many models used in the literature using continuous form or even delay differential equations, just assuming that since you have very many compartments, you don't know how many, you just approximate them by a continuum. And this is also an interesting aspect, because then the question is, are these models completely equivalent? And we can just choose what we like more or what is more convenient for our work? Or is there anything important or principle in the continuous or discrete structure of such system? And for this, I would like to show you continuous version of the model, which as I just showed you. So now you see, now here you see the equation. So now the very first continuous model for this, how we put, describes first this first compartment of stem cells exactly as we did it before. So it means I have stem cells now denoted by W. They divide at the proliferation rate PW. We have two daughter cells. With probability A, they stay stem cells. Mother cell does not exist anymore. And then you have transition to the progenitor stage, and this we assume to be continuum. So it means there is differentiation along a line, along an interval. And so first we get flux from the stem cells, which will be here defined in the boundary condition. So we have some flux here G, some flux from the stem cells, like the stem cells W. So now if with probability A, they stay stem cells, then means with probability 1 minus A, they differentiate. So this is then description now of this, let's say, of the boundary of our continuous compartment. And what happens in the continuous compartment? You have transport through this compartment, so you have maturation. And then, of course, you have division, and you may have death, if we consider here death also in this compartment. And then at the end, you jump to last compartment, which are mature cells. And again, why they are described now by extra ODE, just because there are different processes. So if I want to be in a classical setting of continuous functions, I mean, and not to have here any measures Diracs, then of course I cannot put it into the same equation. So we put it to an extra equation. So we have again jump from this continuum to mature cells. Now we have flux from this, and then you have death rate. And this model, this continuous model, then this can be parameterized. So the function, the maturation function can be chosen this way now, depending on our self-renewal and regulation, that this model is exactly continuous extension of the other one. Or differently speaking, if you discretize this model, you will get the other one. So one could think they are equivalent. But then you start looking at the analysis of this model. And again, as previously, so now if this coefficient, so if A is smaller than 1/2, then this coefficient is negative, then W must go to 0. If W goes to 0, then everything goes to 0. So if my coefficient, this bracket, which I called now alpha, is negative, then I have exponential extension. If it's larger than 0, then I have solutions which are banded away from 0. This can be shown just manipulating with the equation and just using some estimates from below of these equations. So somehow we have this kind of alternative, which is pretty typical for different nonlinear structure population models. So we are of course interested again in this case when solutions are positive. So it means this is the case when self-renewal of stem cells is larger than 1/2. So exactly like in the ODE case, we expect now strictly positive equilibrium. And this we can compute. A bit maybe more computation than in case of our ODEs, but somehow we set our equations to 0. We again found now the value of mature cells as a function of self-renewal of stem cells. And then just since from the steady state of our transport equation, we get just this first order equation in X, which we can also solve. And then we get the solution of this in a steady state. So we have positive steady state, which is unique. So you see already first interesting issue. The model, although I have infinitely many, so to say, possible stages, the model behaves like two-compartmental model. I have 0 steady state or strictly positive. I don't have all these intermediate things that it could be 0, 0, 0, 0, 0, and then starts suddenly to be positive. So the question is this continuous model behaving like two-compartmental model? And now again, we can look at the computations, which again, I don't put you here because they are just technical and I would say rather boring, just as a maybe kind of summary. So first thing you can think intuitively, if I would have linear transport, so it's like delay. So if it's slow enough or if delay is big enough, you expect oscillations. And indeed, we can show that there is here bifurcation under some conditions, and we can have oscillations. Okay, but one can say it's not my model. My model has this regulation. But even for this regulation, we can prove, similarly as we prove for free compartments, Hopf bifurcation, we can prove also here that there is bifurcation possible, and we will have oscillations. So it means that the system behave in a different way than ODE system, because it has structure of steady state like two-compartmental model, but may have more, but may exhibit also oscillations. And the question is, can we obtain this continuous model from the rigorous limit from the discrete model? And the answer is that in fact, not really, because about this rigorous limit shows us where is the problem. The problem is that to, let's say, put artificially infinitely many compartments, you need to assume that the distances between them are epsilon, are small. And distances between the compartments in our assumptions are related to cell divisions. So you would need to send proliferation rates to 0 or just to rescale things. And this shows that, in fact, it does not make too much sense. So in fact, continuous model make only sense if differentiation is independent on the proliferation. And this shows also first interesting issue. And then one asks, okay, can I build now such a continuous model that could have this discrete points where I could have, for example, this extinction on several few points so that it could be described on the graph? And the answer is one can do such models. We did it quite many years ago, and but these are just technical and sophisticated because you can put structured population models on such a structure with the single points. But then to be able to have semi-extinction of population in these points, you would need to have very sophisticated velocity of this transport, having some non-Lipschitz zeros here and any of such crazy things. And of course, mathematically, it might be fun, but it is not really a good description, in my opinion. But I show this because later, not tomorrow, on Thursday, I will come back to unifying now new approach, which does not need such sophistications to have combination of discrete and continuous points. So this was a healthy system. And what we did, we looked at the discrete and continuous description. And in fact, again, we can parameterize the continuous so that it describes the same as discrete, but somehow at this stage, one could say discrete description is more useful because it is in fact more rich. At the same time, so less technical and describe our processes well. So that's why for now, today, we stick with the ODE description of such lineage. And then we ask what happens if we want to model additionally cancer cell. And again, blood is a fantastic system because in many of cancer models, you just focus on the model of cancer cell population without thinking about healthy cells. And in fact, in some like brain tumors, you even cannot think at the same time about healthy cells because they don't divide or things like this. In blood, the two systems coexist. Cancer grows in the system which is still producing healthy blood. And the problem is, of course, if there is too many of this leukemic cells. So, and even more recently, it was shown experimentally analyzing bone marrow that if leukemic cells number grows, then at the same time, number of healthy cells decays. So this is in fact one of the problems of leukemia, that healthy system goes down, a healthy part of the system goes down and it's not able to perform the function. So that's why in this system now we have the two lineages which must be coupled. And we are interested in understanding how this healthy and cancer cell lines are coupled. And yes, this means also how to describe them together. And of course, first, let's say, ad hoc idea is I had before my model of healthy line. So now I assume I have cancer cells and they may respond to the same feedback. And now I even put these equations, but we will not look at them too much. Just you see, you may say, okay, we have leukemic cells, we have healthy cells, we have these different compartments for leukemic cells, for healthy cells, and all of them respond to the same signal. So as in our healthy model, self-renewal is multiplied by the signal intensity. And now just signal intensity couples these populations which are in the bloodstream of cancer and leukemic. Okay, so this is the model. One can also ask questions, okay, so why do we assume that leukemic cells respond to healthy regulation? Maybe they just ignore it. They don't respond to cytokines at all. They don't need them. But maybe the only regulation in the bone marrow is competition for the space, as in many other cancers. So it would mean that now in my equations, I don't have in cancer line all this signal intensity coupling. Instead, my death rates of the cells which live, okay, this first part in the bone marrow are regulated, and they are regulated just as in logistic equation or things like this, just by the total number of the cells. So I may have such two models. I will not now discuss at all details and not compare them, but I can tell you that it's interesting because mathematically these equations have in fact, very similar structure. Of course, they are differently written in terms of nonlinearities, but in terms of dynamic and structures, it's pretty similar, although there might be also some differences that might be interesting for applications. So we also had such work, but just we don't need it now. So we focus on the model with regulation through cytokines, and again from very simple mathematics, just linear stability analysis, we get condition for the destabilization of healthy cell system and growth of leukemic cell system. So it is like we were in steady state of healthy, we introduce few leukemic cells, and we ask under which condition this healthy lose stability and decay, and this leukemic overtake and growth. So simple question, and we may get condition even for bigger system. We get condition which in fact involve stem cell parameters. And you see this general condition involve self-renewal, involve proliferation, involve death rates, and one can find different combination where the cancer cells will have this this inequality comparing to the healthy cells. This includes, for example, the classical interpretation, then you need increase of proliferation and decrease of cell death of cancer cells. But this will not always give you loss of stability. But what always works, if self-renewal of cancer cells is larger than self-renewal of healthy cells, cancer cells win and growth. So again, shows importance of the self-renewal parameter. And this was also interesting because somehow when we found that with this very, very simple mathematics, it was still the time when our clinicians, they did not think this way. So somehow you see that even simple mathematics is sometimes ahead of experimental knowledge. Of course, nowadays they already showed that experimentally that self-renewal of leukemic cells is higher than of healthy cells. Okay, can we then focus just on two compartments and look only on leukemic cell parameters? The question is yes, and we tested it also computationally in the following way. You may take a model with multiple compartments and then just look how perturbation of these different A parameters and proliferation parameters induce growth of cancer. In particular, we look how fast your tumor mass is growing, because it's important, because you have detection of the cancer when there is, let's say, sufficiently many cancer cells, or differently speaking, sufficiently small amount of healthy cells so that the system is not working properly and the patient search for diagnosis, search for help, and then disease is diagnosed. So here you see how in the space of proliferation versus self-renewal values, what is the relative time till the cancer mass achieves some size. And you see that in fact, if of course they would be very small, then it's very large time for stem cells, and if they are larger, then this times goes declines very quickly, and you see here very big differences in these times. While if you do such manipulations with parameters of intermediate compartments, you see hardly any difference. So it means, in fact, forget about n compartments. We can work with structure of two compartments and focus on parameters of cancer stem cells. And what we can do now with this. So you see now, if I know the time to diagnosis, then I can get from here where I am with my parameters. Okay, I don't get unique values. I get a curve on which my parameter self-renewal and proliferation lies. And as if you remember the equation always A is multiplied with P. So it's clear that I get this A times P as a value rather than any of them initially. So, but this means I can estimate parameter if I have some numbers, but if I also have this time where I know my changes in the size. And if you have first diagnosis, you don't know how much time it took. But in case of acute myeloid leukemia, we have patients with multiple relapses. So here you have two examples of such data. So the patient had some measurements of leukemic blasts. Then he got a chemotherapy. Then the blast number went down to 0. And then after a while, there is again growth. Or here, even with multiple relapses. So we can use data of such patients to try to understand where their cancer stem cell parameters are located. So these are parameters which you could never measure experimentally, especially self-renewal. So, and then we did it for, I think, 31 or 32 patients. And for each patient, we found such a curve in this space of parameter self-renewal versus proliferation, describing his dynamics. And this, you see also the data are pretty bad. I have just few points because they are data from patients. But still, even for such data, we could get these curves. And then what we can do, so we see that the position may change, like going more or less in this direction of right upper corner. And you can do statistics on this, comparing with the outcome for the patients. Usually we know, we have the data of the patients about whom we already know what was the survival time. And here it is significant the localization of this curve is statistically significant for the prognosis or for the survival of the patient. We could really split them. And even more interesting, if you have patients with multiple relapses, the curve moves in a bad direction. So somehow it seems that using such simple models from patients' data, we could estimate parameters of cancer stem cells which are correlated with patient prognosis. Okay, so this was the model of leukemia. And now we would like to understand more, I promised you, heterogeneity. So first, of course, these days, there are more and more studies with this single cell data, which I told you about at the very beginning. But first of all, not all the time they can get them. Getting single cell data are very expensive. And also, let's say, historically, the first insight into heterogeneity was through looking at some population genetic analysis, but of the population data. So what you can get in such data? So you get information about percentage of different clone. And what is a clone? So if I have mutation A, and then if I have another mutation, so each daughter cells of this mutation, if there is no other mutations, will have this mutation present. So it means if I had, for example, here 1, 2, 3, 4, 1, 2, 3, 4 different this different mutation structure of the population of the cells, I may have here daughter cells of the cells, and I will have 4 clones. So this is the clonal structure of the cancer cell population. And there were 2 Nature papers, 2011, 2012, showing that for leukemia, in fact, you don't see so many clones. Usually you see just a few clones, like 4 or maybe 5 or 3 clones for the whole leukemic population from a patient. Moreover, it was shown that sometimes if you look at the diagnosis and you look at your clonal structure, sometimes at the relapse you see the same clones. They could have different percentages now. But sometimes it might be that a clone which you saw before disappeared, or it might be also that new clones appear. And the question is, can we describe this clonal dynamics? In particular, can we describe clonal selection, so disappearance of some clones? Or can we explain appearance of some clones without additional mutations on the way? So for this, we extended our leukemic model. So before I had this healthy line, and leukemic I. And I already reduced them to two compartments. Like I have dividing cells and non-dividing cells. And now we say, okay, we have not one line of leukemic cells, we have different clones. So we may consider different clones. And if I have different clones, I may parameterize their cells with different A and P parameters. Okay, so I have now such a structure of my model. And we can study it with, again, with ODEs, if you want. And then you can look at the specific clones that even without any intervention, there must be clones which grow but then decline, while other clones come later. And so it means depending on when you have your measurement, you may have different structure of this population. And now if you, maybe this I like say later. So now the question is, can we understand this dynamics, or in particular, can we say which clones will be selected, will survive? And for this, we took a continuous version of this model. This is just mostly, I can say, for nice mathematics or for elegance one. Of course, if we have results for this continuous model, I could reproduce it for the ODE just with, which would be maybe not so nicely written. So what means continuous version? So now I'm still working with two compartments, but instead of having healthy cells and N clones, I assume now that my heterogeneity with respect to parameters is continuous, is continuum. So it means I put this heterogeneity on an interval, for example, 0, 1, when in 0 I have healthy cells, and then I have different clones. And you know, this is now denoted by X. So it means now my parameters, self-renewal and proliferation, depend on X. One could also say my heterogeneity is the pair A and P, and then one could consider two-dimensional structure, but still seems to be easier to consider one-dimensional structure, and then parameters which depend. So it is like before we had just n+1 points, now we have an interval. And then we can rewrite the equation. So now instead of writing n+1 equations, times 2, because I have two compartments. Now I have just 2, so to say, times infinity, because I have my continuous structure. So I have U cells, these are these dividing cells, and this equation looks exactly like my stem cell equation before. So again, 2 daughter self with probability A stay, A is regulated, mother cell disappears, I have proliferation rate, and then 1 minus this A is comes to the second compartment, and then I have some death rate in the second compartment. Okay, so again, the difference is now that they are structured by X. So it means my parameter A depends on X, my parameter P may depend on X, and then I have the whole distribution of cells along X. So if I would like now to have all mature cells, all cells from bloodstream, I need to integrate them with respect to X. And this I denote by rho 2. So this is the total mass of all my cells, leukemic and healthy, in the bloodstream. And this is what we assumed before regulates our parameters. So now instead of having here just this last compartment, we have here a total mass from the last compartment, the blue thing here on the picture. Okay, so this is the model. So now this is integral differential equation, or one can say differently that this is like a evolution differential equation, but with a non-local term here. And if you, I don't know if you also had heard anything in your lectures about this mutation selection models, which also a lot of mathematics has been done. for last, I don't know, 10 years or more, or even more, then in fact it reminds you already on the selection, on the basic selection models. So I have here non-local term, and you can think like thinking about first equation. I have some value in the bracket, and then I have this proliferation rate. Then it's clear that who has large proliferation rate will be growing quickly, like this blue guy here. Yes, big P makes me growing fast. But at the same time, if these guys are growing, the total mass of V is growing. So this is growing. This is growing. So it means at some moment, this bracket becomes negative. So if the bracket becomes negative, this starts declining. And now I have, for each x, I have different value here because A is different, but they sense the same competition, like competition is due to this integral. So it means those with high A will be still growing, while those with small a will be decaying. So even once see it, I would say quickly, intuitively looking at this equation. But just proving it's a little bit fancy. But what we can prove? We can prove exactly what I said. It means that if my A function has somewhere maximum, that all populations with A smaller than this maximum will decay to zero. You see here the simulations, it's one-dimensional X and then time this direction. And we start with distribution like this. You have a lot of, for example, healthy cells, and then less and less and less of these cells. But then the highest self-renewal is somewhere in the middle. And you see very quickly, everything decays exponentially to zero, while only in the trait corresponding to this maximum of A, we have survival, we have selection, and then we have here the whole mass concentrates in this point. And of course, you have also, this is still nonlinear model, so you have dynamics of this mass. So we may prove this theorem. And again, since I think it's still pretty instructive for the system, I just tell you again. So what I need to prove something like this? First of all, I want to know that the total mass of the system behaves well. But to write equations for total mass is enough to integrate two equations. It's still not an ODE because, of course, here I have this whole expression and here I have A. But for these equations, we can show boundedness and strict positivity of rho 1 and rho 2 with some manipulations. And then we can show that, in fact, everything except of this maximum point decays to zero. How we do it? We just take values into different points, and we can make really point-wise estimates showing that, in fact, the dynamics depend on values of A in this point. So if somewhere A is larger, somewhere else, then in this value, everything will decay to zero. And this way, we can show convergence of the solutions to Dirac measures. Dirac measures because my mass does not disappear. As I said before, from equation for masses, I can show that mass is strictly positive. So mass is strictly positive and everything decays or disappears except of this point of Lebesgue measure 0. So then I have a Dirac measure. And in fact, we can even show that it's not only concentration, but the mass. You see, even for a while, you have dynamics of the mass here, but this mass converges to a steady state. And this steady state is, in fact, given by the solution to equation with parameters corresponding to the maximal value of A. And the proof is also interestingly simple because, in fact, one can prove that if you have a system of ODEs which has global equilibrium, a convergence to global equilibrium, then if you perturb it by functions which for which you can show that they are sufficiently small, like here this are L1 functions in time, and then you have what means at the end that there is a certain decline in all these functions. Then you may show that in fact there is the same equilibrium. And this can be again shown by manipulation of this equation, because we can rewrite our system with the system of the parameters corresponding to the maximum as into parts, and we can show that one of this part is very small. So somehow we proved rigorously, independently of the number of compartments, that we have selection of the clone with highest self-renewal of cancer stem cells. And of course, it means if you wait infinitely long, there will be only this one selected, or if there are several with the same fitness, then there will be several of them. Here for finite times, both in simulations and in the data, you may have also other clones still surviving for a while. And we showed that you can reproduce these experimental patterns by simulations without introducing new mutations. Because it is sufficient that some clones exist before, just they are so small that you cannot detect them. So, and this model with multiple clones could be also compared to patient data, and we could use it to try to find out the let's say clonal structure. So which clone was present, or how clones change, like that some clones decay, some clones appear. And it is interesting, theoretically interesting potential application is that sometimes clinicians know what is the mutation. They have all these fancy names of mutation, but they don't know what this mutation means. If this mutation means exactly increased proliferation, increased self-renewal. So now we could estimate parameters and find out how the mutation can be linked to a characteristic of cell populations. And then, of course, if we understand which mutations govern dynamics of cancer, like which cell properties govern dynamics of cancer, we could think about efficient therapy. And then we also could extend it to the models with this, because I said, okay, everything is reduced to 2 compartments, but you could also extend this structure not only to n compartment, but even to continuous model, and show in fact that these intermediate compartments, they don't play any role. So somehow it's always about cancer stem cells. And the last thing I would like to somehow tell you about in this context is that again, if you look at the selection models, it's always also that people consider selection mutation model, where you have additionally mutation term. One can say, okay, I proved that we have a selection of one clone with maximal fitness, but if there are no mutations introduced on the way. So now the question is, what if I have these small mutations? And usually you model such small mutations or with some diffusion, or you can model it with this kernel operator. So okay, here for several reasons, also for the fact that later I want to work with just more measures than continuous functions. It seems that this kernel description is, let's say, more convenient, but at the end they are equivalent. So you can of course here consider now this mutation kernel with compact support, what does not change the fact that that the mutations will propagate through in such PDEs anyhow for the whole line. And these mutations are scaled now with small parameter epsilon. And that's why also you have this epsilon here, because it means that not only after division mother cell is not there, also epsilon fraction of the new cells is not there because they mutated to a different clone, different trait. Okay, so we have now our old model for which we know that there is selection and to which we add the mutation term. And what we can prove here? We can prove that stationary solutions of this system converge to Dirac deltas if epsilon tends to 0. So, question is what it really means. So first of all, it means that we look at stationary solutions. And then it means that if we have here this mutation smaller and smaller, then we are closer to this selected clones. So okay, we can say if I have a small epsilon, I will have smaller, like more concentrated solutions. But you already see here also that in fact it's not completely satisfactory because you would like to know what is the asymptotic of the solutions of the complete model, and now we somehow assume that we can look at the stationary problem. But what we can in fact shown we can show. I mean, I put it on the slide, but again, just maybe even you should not try to read it. This is like an idea for, so I mean, this model is pretty tricky because you have this crazy nonlinear growth. And how you can treat this model? Again, the first equation is only needed, like this is the equation for the mature compartment, since we have mature compartment in the regulation. So we can even integrate this equation and consider it already in terms of rho. So I have a model, this is my equation, and I look at the stationary problem. And I want to show first that I have a solution of the stationary problem. So here one can do it just reformulating this as a kind of eigenvalue problem. So namely, you can think that that you have an eigenvalue problem and you want to find that you have eigenfunction corresponding to this eigenvalue 0. And then if you think, and then you want to have this stationary distribution. So you want to have the positive stationary solution. So somehow if you think it is at the end brings us back to the theory which was first well developed for ODEs and then in meantime also extended to many different structured population models. So it means the theorem, the application of Krein-Rootman theorem that gives us under certain assumption on our parameters, that gives us that there exists a positive eigenfunction and this principal eigenvalue. And here somehow we can again reformulate the problem that instead of looking at this problem when we search for 0, we reformulate it as eigenvalue problem and we search for this eigenfunctions corresponding to the eigenvalue equal to 1. And this can be here shown. And if this can be here shown, then we have the stationary solution. And then for this stationary solution, we can set epsilon to 0 and show that the solution concentrate. And then we can also look at the linear stability of the whole system. But again, it is not the same as showing full dynamics. But why I showed you this, first of all, that's just to show you that also in case of this leukemia models, mutations will not change the dynamics. But the other thing is to show you that if you do it with all these classical settings, you find quite a few of nasty things. First of all, we were in the model maybe with some continuous, I don't know, regular solutions, then suddenly everything should converge to Dirac measures, which are not regular solutions anymore, of course. So in which sense it converges? So we can, of course, always think about this fancy functionality setting that we have this convergence in some weak star topology or things like this. But everything is very unfriendly. Then, of course, probabilist would tell us, so then consider convergence in probability measures, but our distributions are not probability distributions because we have growth in the system. So somehow what I want to say with this, that you see that if you put such models in a classical setting, you have many mathematical places when the things are not elegant and not cleaned. And this motivated us to look at the more unifying and elegant mathematical setting where we don't need to worry so much about what it means for me that my solutions converge in some weak star topology. Yes, so like we prefer to have everything in some sense metrizable and considered this way. So I think that this is when I just don't skip this for today. I just want to finish this part of blood modeling, of some summary of this part. So first of all, even based on this, let's say, more classical experimental settings from last 10 or more than 10 years, we can already observe that we have heterogeneity of cancer populations, clonal heterogeneity, but this heterogeneity, it is not just there, it has also dynamics. So we may observe clonal selection, or we may observe emergence of heterogeneity during mutations. And mathematical model provide us a tool to explain and understand if the things are consistent and what all these features mean for the disease dynamics. Clonal hierarchies allow conclusions about properties of individual clones on the level of cancer stem cell parameters, which cannot be measured experimentally. And estimated cell properties differ between different patients, and they are significant for the dynamics of the disease, dynamics of different populations for different patients. So from this perspective, it seems that such models can have also a prognostic character and could be correlated to individual patients. So they could have potential for this individualized medicine for patients. And what is interesting, that the models are still very simple mathematically and could be implemented in a very efficient way. So on Thursday, I would like to show you even different types of result if you look at the heterogeneity of neural stem cells. And so in the case of neural stem cells, we'll look more from experimental perspective than from the clinical perspective. But somehow looking at all these clonal structures suggested us also recently new approach to looking at glioblastoma, which I also would like to show you. And I would like to show you, let's say, more mathematically elegant framework to treat all these evolutions, which may concentrate or may start from concentrated, and somehow framework which allows us to include Dirac distributions and different or discrete structure of the state space combined with continuous without let's say without forgetting about mathematical elegance. And here I would like just to acknowledge my collaborators from this blood stem cell modeling about which I was talking today. So most of the work was done together with Thomas Still, who was my PhD student and postdoc, and now is professor of computational medicine in Aachen, but also with another PhD student, Jan Erik Busse. We collaborated with clinicians, hematologists in Heidelberg, and also different parts like more mathematical, analytical were done with our friends from Warsaw, Torino, Barcelona, and Paris. So thank you. I think Thomas is not there anymore, so I don't know if you have any questions.
I have a very naive question. You use row 2 at the denominator, but what about row 1? It's not playing role in the denominator of the equation for U1?
No. And this was strongly motivated by applications, by this application. And this you see it in the very first models. If you look at the ODE models already of blood, because what we say, or maybe, I don't know which equations, what we say that parameters are regulated by signal, which is, let's say, governed or regulated by cells which are in bloodstream. And cells which are in bloodstream are these cells, or maybe these cells. So this is our, let's say, in the two-compartmental version, second compartment, and not these cells. These cells are sitting physically somewhere else in bone marrow. And in this model, the total number is not having any influence on the regulation. You may have it. That's what you maybe also thought about. If you look at the model, where I have it. If you would look at this model, what I told you, that one can alternatively consider the model when all cells sitting in the bone marrow compete for the space. Then, of course, in the regulation, you would have two populations. So okay, here in the discrete model, they are sums. So here you would have two integrals. But this would be then model with a bit different structure. And in this model, you have it only in this feedback from row 2. And in fact, this is a problem, because this is why I could not consider model this equation for any boundedness things like equation by equation. And that's why I also cannot use this Hamilton-Jacobi approach for showing selection and so on, because I cannot control U without controlling V. Yes, you see? So I have to know, if I know that row 2 is controlled by row 1, then I'm at home, because then this equation looks more or less like logistic equation from the dynamics. So this is exactly the biggest difficulty in our work, is to show that that row 1 is controlled by row 2 is controlled by row 1. So we need such estimates. If you look at the equation for rows, in fact, you then look at equations for the quotient row 1 over row 2, you show boundedness of this quotient. And that's why you know that this, if you do estimates from above, then you want row 2 to estimate from below by something times row 1. And then if here you will have row 1 and not row 2, then the things became much easier. So, but the problem came from modeling, so to say.
Okay, what about the coupling of healthy and unhealthy cells?
Mm-hmm.
It's happening linearly or non-linearly?
Okay, so here it's always
Is this very simple or very complicated?
No, so dynamics is, because row 2 consists of healthy and cancer, yes?
Okay.
Row 2 is all this blue together. So it means healthy and non-healthy, just those who don't divide. So it means healthy and not healthy, you have them, because somehow in this model without mutations, you would think you have them in parallel, yes? Because for each X, like healthy and not healthy, it's like here. That means for certain X, you have healthy, And you don't have here any, I don't know, diffusion operator. The only coupling operator in this X space is through integral. But indeed, integral is taking us all this. So this is just non-local equation.
Okay, if you have more time, the stemness role with the division and proliferation, it looks like it goes in different direction, right? If I have low stemness, I have a lot of division and proliferation. And if I have high stemness, I have low division and proliferation, right?
I mean, here the two parameters are not really correlated. So
No, I'm actually relating to the picture that you had, where is some kind of knobs in the, it looks like triangle, long triangle of stemness, high stemness, low stemness.
No, I don't understand what you mean.
Okay, I'll think about it.
Oh, this.
No, it's not on these results. It's the very beginning.
Because
when you have you have some equations.
Yes.
No, not this one. It's much earlier.
Much earlier, okay.
Earlier state. Even you had it already. You go very fast. Okay, we can forget it.
Here I have Lapunov. No, I'm afraid I didn't, because you said I changed P and A. Yes?
No, I'm just interested in the role of stemness.
Yes.
Okay, and stemness can vary.
Yes.
So you had this triangle that you had that picture, but you go very fast. But here. You see at the lower part of both pictures, you have this stemness.
This you mean? Okay, yes.
Of course, it can be understood in the equations, but I want to understand this different behavior. High stemness, we have low division and low degree of differentiation, right?
Yes, so yeah, actually, it's clear that high stemness is low degree of differentiation, because high stemness means that self-renewal is big. And differentiation is 1 minus self-renewal. I would say division can be still decorrelated. That's why I thought that you were speaking about this picture, because somehow this is important. On this picture, you see, like, let's say this picture, you see that if I estimate parameters, I find that parameters lie on the curve. So it means, and the point on the curve shows me already that self-renewal is relatively high. And of course it can, so, okay, I know. The same dynamics here is given by PA from such a curve. So it means indeed, I may lower proliferation rate, increasing self-renewal, and I will get exactly the same dynamics concerning yes. Yes, because I can estimate A times P. Yes, so this is indeed. But this is in fact still very high. So somehow it is not that like if I'm here with P, then I'm here with A. I'm still in this corner, but still indeed I can move one parameter down, the other up. As long as I'm on this curve, I will reproduce the same dynamics. But so I don't know if we think if it has biological meaning.
It can help.
Yeah, the only what I have think here always in this context is that if you, because you know what you can do, you can always kill cells which are fast dividing. So then the question is, if I am here, if I would have fast dividing cells, I could kill them, but it could be also that I'm rather here. And this is exactly what I expect, that in the mutations, they increase self-renewal and not necessarily increase proliferation, because you kill fast dividing clones.
Okay, so
But
there is...
mathematically, it is consequence from the fact that you have always in equations this A times P.
Right.
Mm-hmm.
Okay.
Mm-hmm.
Okay, thank you very much.
You're welcome. Okay, good. So if there are no other questions, so then I think, and on Thursday we already know that the time difference is now such that we meet at 2 hours earlier than today.
10 our time. 10
Yes.
in the
Time, so it means 4 p.m. my time and not 6 p.m. my time. Yes, exactly.
It's better before dinner.
Yes, yes. But it's also better for me, you know.
Of course, that's the idea. It's
Good.
after breakfast for us, before dinner for you. Thank you
Yes.
very much.
Thank you very much. Thank you.
Okay.